Okay, so we are continuing our conversation of meiosis, taking a deeper dive and understanding what would happen if meiosis happened incorrectly. So this is lecture 26, and it has to do with this, I don't know, yeah, it is a phenomenon because we don't really know why this would happen. So this phenomenon called nondisjunction. Okay, so nondisjunction. And nondisjunction leads to something we call aneuploidy, which is the abnormal number of chromosomes in a given cell. So we'll just say abnormal number of chromosomes in a cell. Anu meaning different, ploidy is how we describe chromosomes. And then this process called gametogenesis. So gametogenesis really is meiosis, right? But we're going to get a little more specific and talk about the differences between when ova or eggs are made versus when sperm are made, because the process they are made by meiosis, but meiosis happens slightly different for each of the gametes. Okay, so what if meiosis occurs incorrectly? So a quick recap because we just did this. But if segregation, meaning the pooling of chromosomes, happens correctly, this is what it should look like. So here's your cell that it looks like it's in metaphase, or let's see, what is this showing? Oh, this is just your germ cell, right? So this is your germ cell here. So it's a 2N germ cell. Looks like 2N equals 4 germ cell. Okay. And recall that the chromosomes, if they're not in cell division, they do not look like little butterflies. They are not linked up like that. They're floating around like spaghetti noodles, all random and crazy. Okay, but in meiosis 1, what's happening to these chromosomes is they're supposed to, the tetrads are supposed to split. So here, we know that, let's call this tetrads will form in prophase 1. And then in meiosis 1, the tetrads split, right? So you can tell that this cartoon is showing us anaphase, right? They're being pulled apart. And then so keep going. And then you get to meiosis 2. In meiosis 2, those sister chromatids, pairs of sisters split. So in meiosis 1, tetrads split to form pairs of sisters. In meiosis 2, anaphase 2, the pairs of sisters split. So we say the pairs split to create daughter chromosomes. To create daughters. And those end up going after telophase and cytokinesis 2, those end up going into gametes, where every single gamete has one chromosome number 1 and one chromosome number 2. Every single one of them, but they are not identical. Okay? If it was human, you'd also have one chromosome number 3, one chromosome number 4, ... one chromosome 23. Normal meiotic division, gametes. What if it happens incorrectly? So it can happen incorrectly in 2 very important places. And this is going to happen pretty much like, again, it's metaphase and anaphase, because in metaphase what happens, we know that the kinetochore microtubules are supposed to attach. If we're talking about meiosis 1, they're supposed to attach to the entire pair. The kinetochore microtubule attaches to the entire pair, and then in anaphase, it splits apart. So if meiosis does not happen incorrectly, it can happen in metaphase 1 because the kinetochore microtubules did not attach correctly. So what does that mean? They should be attached. So I'll just draw a quick cartoon. They should be attached. Let's draw the synaptonemal complex so we know that it's still there. Okay. They should be attached to the entire sister pair of a tetrad, one pole and the other pole. Okay. If it did not happen incorrectly, if it did not happen correctly, so this is correct. Incorrect would be, here's your chromosomes, let's draw them again. Draw our synaptonemal complexes there. Incorrectly means that kinetochore from one microtubule attaches, and then no microtubule attaches to the other side. Okay, so there's no microtubule coming from the other pole. That's not good. So when these kinetochore microtubules shorten, so if this is shortening, it's just gonna yank the entire tetrad, this whole thing, even though it may not be attached to both kinetochores, it's attached to one. And this chromosome is attached to the other because it's zippered up. So if you're just attached to one of these and then you start retracting your fish line, you're gonna retract and you're gonna pull in two fish. Imagine like one fish is biting the tail of the other one and its teeth are lockjawed. That's it. So even if you have only literally sunk your hook in one fish, because that fish is attached to another fish, you're pulling both of them. Okay. And if you pull both of them, then both of them go to one cell and the other cell gets no chromosome number 1, right? So let's get our numbers in there. This is chromosome number 1. So this is how it should look. Instead, if meiosis metaphase 1 happens incorrectly and the kinetochore microtubules don't attach, after the first cytokinesis, what you end up having, so the first, I'm going to draw it right here. So the first cytokinesis, what you end up having is you have this one happens normally, great. Okay, and let's draw the one that did not happen correctly. And these are still attached via their little synaptonemal complex. And then you have nothing over there. So you have your chromosome 1, and chromosome 2. No chromosome 1 over here, chromosome 2. So we say no chromosome 1. That's not good. There's thousands of genes on a single chromosome. So usually this is not viable. The cell will recognize it and will undergo apoptosis. It will just kill that gamete. But sometimes the apoptotic mechanisms don't happen when they should. And so that gamete possibly can survive. That's going to be an issue. Okay, so let's continue. So you can see here that both of these, this has an issue and this has an issue. So when this cell undergoes meiosis 2, all of its products are also going to have an issue. This has an issue to start with. All of the products are going to have an issue. It's like if you have a problem at the top, that problem trickles down no matter what. So if this cell underwent the next level of meiosis, what would happen? Well, we know that these are now going to be in a single cell. So here they are. Okay, so let's draw all these. So we have a number 2 and number 2. Oh my God, we have so many chromosome number 1s over here. So many. Okay. But these sisters are still going to be, the pair is still going to be pulled apart. And you can see, right? You can see what's going to happen. The left half is going to go into one gamete. The right half is going to go into another gamete. And after telophase and cytokinesis 2, what you end up with are gametes. This gamete has 2 number 1s, right? Oh, I guess 2 chromosome number 1s. Last semester I called that chromosome number 2. Okay, so we say that the gamete is disomy chromosome 1 or disomy 1. Di meaning you have 2 of them. You shouldn't have 2 of them. You should have 1. Right? And so both of these gametes are disomy 1s. They have 2 chromosome number 1s. This is a no-no. This one has no chromosome number 1, 0 of them. We don't have a word for it. It's not monosomy. It's just missing chromosome number 1. These are the gametes that are not viable. These are these are probably not going to be viable either. If you're missing a total chromosome, most times, so we'll say 99% of the time or higher, this will lead to apoptosis. Having extra is not a good thing either. So these are also made incorrectly, but this doesn't always lead to apoptosis. Most of the time, it's not viable, mostly not viable, meaning you can't create an embryo with it. Most of the times it's not viable, sometimes it is, and if that gamete is used to fertilize, to create the next generation of embryo, that embryo generally tends to have some developmental disorders or delays, or the development in utero is so bad that the embryo gets aborted. Okay, so usually it's not. We have systems, we have checks and balances for this, but sometimes you can still make an embryo. So they're mostly not viable. Most of the times they will become, they will undergo cell death as well. Okay. So if it happens early, so we said in this case, the incorrectness was happening in meiosis 1. Why? Because the chromosomes did not attach correctly. Well, guess what? There's another metaphase where the kinetochores better attach correctly. And this is another place where meiosis can happen incorrectly. So let's see what happens there. So as a recap, when it happens, so this, oh my gosh, can't believe I forgot to say this. This process of not attaching correctly, the kinetochore is not attaching correctly, this is what's called non-disjunction. I don't know why. Sounds like a double negative. So disjunction, junction means, you know, I don't know. I'm trying to rationalize it. I don't know why it's called non-disjunction. So the junction is not happening correctly. I don't know. Anyways, it's called non-disjunction. And so usually non-disjunction will happen, it happens very infrequently. Actually, you know what? I don't know if it happens infrequently. I know that when it does happen, the gametes don't survive. So maybe that's why I think it happens infrequently. So when non-disjunction occurs, it typically only occurs with one chromosome. Rarely, even rarer, will you see non-disjunction happening for multiple chromosomes in a single cell. So you see how it's only happening to this tetrad? It's not going to happen to all tetrads. That happens, like the percent of that is like 0.002, something crazy. It's very, very small chance of that happening. But if it does happen, it's called complete non-disjunction. Yeah, so complete non-disjunction. This is how you can get a male calico cat. We won't go into the genetics of it, but calico cats are always female unless complete non-disjunction occurs. Okay. All right. So if non-disjunction happens in metaphase 1, you end up having both of the cells after cytokinesis 1 are abnormal. And then each of these cells will then produce abnormal gametes. So you can see here that all of these gametes are abnormal. They all exhibit aneuploidy. Aneuploidy, the abnormal number of chromosomes. All of them are exhibiting aneuploidy. Okay. So any of those gametes, if they do make an embryo, will produce an embryo that has the incorrect number of chromosomes. Whether you have an extra chromosome number 1 or only one chromosome number 1. Because these are the gametes, so you can assume that the other gamete has the normal amount. Let's look at non-disjunction in meiosis 2, metaphase 2. So this first thing happened correctly. So the tetrad gets split, great. And then the tetrad, the pair of sisters should get split. So in this case, again, kinetochore microtubules did not attach correctly. Okay, so non-disjunction is happening in meiosis too. So remember, in metaphase, you should have a kinetochore microtubule attaching to each of the sisters so that in anaphase, those sisters get pulled apart. If the kinetochores did not do that correctly in metaphase, then the same thing happens, that only one pole extended a kinetochore microtubule and there's nothing coming from this side. So once in anaphase, once that kinetochore microtubule starts to shorten its line, it's going to bring the whole pair with it. Okay, so that's still a pair. That's not supposed to happen. Right? So this is going to only happen in one of the cells, right? Because if this meiosis 1 happened correctly, it's going to make 2 correct cells. So I'll draw it right here so we can remember. So if this happened correctly, what would you have? You would have a cell that has a chromosome 1 and a chromosome 2. And then you'd have another cell that has a chromosome 1 and a chromosome 2. Okay? That's what would happen. So let's draw the arrow so we know that, let's say this picture on the left is representative of when this cell will actually exhibit non-disjunction, and this cell, meiosis 2, happens normally for it. Again, just reinforcing this idea that non-disjunction, just because it happens to one cell or just because it happens to one chromosome, doesn't mean all of the chromosomes are undergoing non-disjunction. It doesn't mean that all of the cells are undergoing non-disjunction, right? It just so happened that this cell experienced non-disjunction. This cell went around its merry business and did meiosis properly. Perfect. Okay, so we're not gonna worry about this because this happens normal, so it's gonna produce 2 normal gametes. So we're not even worried about this. But in this one, oh man, the kinetochores didn't attach correctly in metaphase 1. So when you pull them apart, in metaphase 2, so when you pull them apart, shoot, one of the gametes is gonna have disomy, chromosome number 1. The other gamete has no chromosome number 1, none, no chromosome number 1. So this cell is going to make 2 abnormal gametes. These abnormal gametes are going to make an embryo. If they're used to make an embryo, they're going to make an embryo with aneuploidy. These exhibit aneuploidy. The future embryo would also exhibit aneuploidy. Because meiosis happened normally here, these gametes will be normal amount of chromosome number. So if either of these gametes go on to make an embryo, the embryo will not exhibit aneuploidy. It will have the correct number of chromosomes. 2n equals 46, where each set has 23 chromosomes. So what you can see here, or what I'm trying to get you to see here, is that when non-disjunction happens earlier in meiosis, then the resulting gametes, all of them, 100% of them, are aneuploids. But if non-disjunction happens in metaphase 2, then anaphase, pulling apart, it happens in anaphase 2, only a portion of them, 50% of the gametes, will exhibit aneuploidy. Why? Because typically only one of the cells will undergo non-disjunction, the other cell will not. So this other cell will make normal number of chromosomed gametes. Okay. So the phenomenon of non-disjunction is the incorrect pulling apart. So this is going to either happen in anaphase 1 or it's going to happen in anaphase 2, but it is the result of the kinetochores not properly attaching, which is why the metaphase to anaphase checkpoint is critical. This is what is checked between metaphase 1 and anaphase 1. Are the kinetochore microtubules attached properly? So those are our checks and balances. And so sometimes the checks and balances, the checkpoint people, they didn't catch the mistake. Okay? But then the apoptotic mechanism is like, oh gosh, that other teammate of mine just completely blew it. All right, I guess I'm killing this cell. And then the cell's bye-bye. Okay, so if fertilization occurs with an abnormal gamete, what's gonna happen? So let's just say all of these in this cartoon are eggs, okay? And so eggs need to be fertilized by sperm. And so this is going to be normal, right? The normal situation. This is when non-disjunction happened in anaphase 1 as a product of metaphase 1. And this is when non-disjunction happened in anaphase 2 for one of the cells, but this one is normal, right? We just finished talking about that. It's the same thing, we're just carrying this stuff down, okay? All right, so let's say the sperm is normal. So let's say a sperm, okay? It also is going to have one set of chromosomes. So if it's human sperm, it would be N equals 23. But in this cartoon, it's going to be an N equals 2. So you have one chromosome, number 1, and one chromosome, number 2, right? Normal sperm, no aneuploidy. So this is aneuploid, this is aneuploid, this is aneuploid, this is aneuploid. So is this, so is this. The rest of them are just normal. This is aneuploidy. Okay. So you can clearly see that if, I'm just gonna highlight this, if this sperm fertilized this egg, the product would be an embryo, right, with a set of chromosomes from the sperm and a set of chromosomes from the egg. And this embryo is now a 2n equals 4. Here's chromosome number 1 and here's chromosome number 2, and there's 2 sets of them. That's what will happen. That's what does happen. And then this is the 1 cell embryo. And then this undergoes mitosis a bunch of times to create a baby. Whatever baby you're talking about, cat, dog, elephant, human, whatever. So it's development. I guess it would be a 1 cell zygote. Okay, no, it's a 1 cell embryo. Okay, so what about when this sperm, a different sperm that exhibits no aneuploidy, if it fertilizes an egg? Let's say the eggs did not apoptose, the apoptotic mechanism fell asleep on the job and let these gametes survive. So if that's the case, I'll just draw one more sperm just to kind of remember what's happening here. Okay, you have, looks like a balloon. Okay. Chromosome 1 and chromosome 2. Now, if you had, for example, this sperm fertilized this egg, what would be the product? The product, you'd have your chromosomes from the sperm, and you'd have the chromosomes from the egg. Okay. So here's your 1 cell embryo. But if we look at the chromosomes that are in that 1 cell embryo, you have 3 chromosome number 1s. You're not supposed to have 3, you're supposed to have 2. Okay? You have 2 chromosome number 2s, that's normal. And if this was a human embryo, you'd have 2 chromosome number 3, 2 chromosome number 4, all the way to chromosome number 23. Okay. So this embryo we describe as tri, because there's 3 of them, somi, because chromosome me, trisomy 1. Okay. So anytime you have 3 of a chromosome when the animal or the organism is supposed to be diploid, we call it trisomy, whatever the number is. Okay. And so if you look at that, if this was the product of egg development, all 4 of these gametes would produce an abnormal embryo. 2 of them would be disomies or trisomies, and then the other 2 would be monosomies. So we quickly draw that. Let's say that the sperm, let's draw it in red. Let's say this sperm fertilized instead that egg that I just circled. So if that was the case, what would the embryo look like? And the embryo, here's the embryo. Okay, so you have the genetic material from the sperm and the genetic material from the egg. So you have 2 chromosome number 2s. Uh-oh. You have 1 chromosome number 1. That's not a good situation because you need both chromosomes. Chromosomes have thousands of genes. You're like, yeah, well, there's one there. I know, but we need both chromosomes. We need them. And it's really because, you know, as one chromosome is being central dogma, the other chromosome may be doing the same thing because you need twice as much protein in that number of time. And if you only have 1, then it's an issue. So you can't go on. I really don't know of any situation where an embryo can be viable if it's missing an entire chromosome. So what do we call this? We call this phenomenon, or this embryo would be monosomy 1, okay? Because it only has 1 copy of chromosome 1. So here we have, in this situation, I'll just draw the lines of this. I know that it's not really staying in the columns that I was trying to stay in, but 100% of the embryos have some sort of aneuploidy. 100%, no aneuploidy. Okay. And then if you had nondisjunction happen in anaphase 2, only part of them, so 50% of the embryos would be aneuploid, 50% normal, right? So if the egg fertilized this, the embryo would be trisomy 1. If the sperm fertilized this, trisomy 1. If the sperm fertilized this egg, monosomy 1. If the sperm fertilized this egg, normal, diploid embryo. This one, normal diploid embryo. Okay. Okay. So, any questions? Okay. So, people have the ability to look at chromosomal integrity, just meaning, hey, I'm interested in procreating and I want to make sure that my future embryo is going to be viable, and I want to make sure that there's no genetic abnormalities. So you can use a karyotype, and that's not the only reason you can use a karyotype. You can also use a karyotype to identify diseases. It's not a lot of information, but I think that this is really, really cool. So karyotype really is a scientific technique. It's something that's done in the lab where you can analyze the number of chromosomes, the integrity, like are the chromosomes damaged? Are there translocations? Are there deletions? You can look to see whether or not chromosomes are present or absent, or whether there are large chromosomal mutations. So how do you do that? Well, the process is called G banding. Giemsa, I think. Giemsa? Okay. So here it is. I thought it was Giemsa. Okay, I'll have to look into it, but it's called G banding. And so remember, chromosomes are going to, like, parts of them are going to be wound really tight, like the centromere. Other parts are going to be loosely bound, like euchromatin. Some are going to be tightly bound, like heterochromatin. And so when they're tightly bound or versus loosely bound, this stain, so you take these chromosomes and you stain them, and the stain will attach better to heterochromatin. Why? Because there's more material there, so it can bind and stain darker. So if you are using this Giemsa staining, the heterochromatin parts of the chromosome, the tightly wound, where there's more condensed in a tiny little area, is darker. And then the loosely wound up chromatin, or the euchromatin, there's less material in that location, so the stain binds less. So it looks like lighter bands. So you basically are creating these banding patterns on the chromosomes. So how do you do this? First, you take your cells and you treat them so that you make sure you get them into mitosis, okay? You get them into mitosis and you stall them in between prophase and metaphase. And you can use a certain drug to do this. And so it stops the cell cycle from progressing. So all of them reach the right between prophase and the start of metaphase, all of them stop there because there's some drug that inhibits them from proceeding. After that, you treat the cells with trypsin. Now trypsin, here's trypsin. Trypsin is a protease, so it's going to start kind of getting rid of all the histones and the nucleosomes. You don't want those in the way, get them out of the way. So you get rid of the nucleosomes by trypsinizing the cells. And it also kind of makes the chromosomes available, right? There's a bunch of proteins in the cell, so get rid of all the other proteins. We're only interested in analyzing the chromosomes. After you have treated the cells in trypsin, then you incubate them in a chiasma stain. So it's just like a solution, kind of like Kumasi glue or whatever it is. You pour the chiasma stain all over your cells and you just let science happen. Wait. And then the heterochromatin will come out dark, the euchromatin will come out light. And then you can arrange this. So now you have to use a computer after this, where you take a picture of the chromosomes, like you actually use a microscope. You take a picture of all the chromosomes, but they don't line up perfectly. Chromosomes don't know how to line up. So you take the picture and then you use a software to kind of drag the chromosomes and organize them yourself. And people that do this for a living, they are able to recognize what chromosome 1 is versus chromosome 2. I can't, because these all look pretty much the same to me. Like if they were scrambled up in a dish, I wouldn't know the difference. But people that study chromosomes and do karyotypes can. Why? Because the banding pattern from person to person for chromosome 3, I guess, always looks like this. So if you took this, even though this looks like it could be the same length as this one over here, the fact that the banding pattern looks like this tells the scientist this is chromosome 3 versus, oh, nope, this is definitely not chromosome 3, it's chromosome 6. How? I don't know. That is insane. But I guess if you do something every day, you are privy to seeing all those differences. So you use your little mouse, you drag and drop your chromosomes, and then you're able to analyze them. And so this is a karyotype, what I'm showing you right here. Oh, come on, don't do this to me now. All right. And so what you can see from this karyotype, there are 3 chromosome 21s. And so 3 chromosome 21s, this individual, I don't know if this came from an embryo or from a human, like a grown adult, I have no idea. But I do know it came from a human because there's 23 of them. So this individual is trisomy 21. And trisomy 21 is one of the few aneuploidy situations where the human can develop and can actually be born and have a certain shorter lifespan, but there are some developmental delays in this individual, and this is known as Down's syndrome. Okay, so trisomy 21 results in Down's syndrome. And then if you look at this karyotype, there's one other abnormality with regard to chromosome number. And this individual also has 2 X chromosomes, which usually humans either have 2 X chromosomes or an X and a Y chromosome. And so currently in the science field, we identify the presence of a Y chromosome as the individual being a biological male. And so this person has 2 X chromosomes and a Y chromosome. So the mere presence of the Y chromosome, this individual is a biological male by definition, but will develop this condition called Klinefelter's syndrome. Klinefelter's syndrome, yeah. And so there are some reproductive abnormalities for this human being. So mostly this human being is infertile. But as you can see, for some chromosomes, aneuploidy does not result in embryonic abortion, right? So the embryo developed to term, was born, and then this person can live out in the regular world. It's fine. Okay, and so you can see the karyotype, the first number indicates the total number of chromosomes, whereas most people only have 46. Then the sex chromosome, so most people either have XX or XY. There are other sex chromosome aneuploidy syndromes as well, so this was just one example. And you can see that there's an extra chromosome 21. Okay, so that's how you would read the karyotype. And so not only can you identify aneuploidy in using a karyotype, but you can also, now that we know more about diseases and the correlations between diseases and genetic abnormalities, some diseases and disorders are linked very closely to chromosomal abnormalities. So CML, or chronic myeloid leukemia, is one of those diseases. And so you look at the results from a CML patient. And so let's look at this here. And this, you're like, what's all that? Okay, I'm going to try to explain it to you. So this little T right here indicates translocation. And translocations, we know, is when two different chromosomes, non-homologous chromosomes, switch material. That's not crossing over, because crossing over happens between homologous non-sisters. This is non-homologous. So chromosome number 9 switched genetic material with chromosome number 22. And then what's the Q and the Q? So chromosomes in human lingo, so here's our big chromosome. Chromosomes, we say that they have arms. So I don't know why we chose the letter P and Q, but one of the arms we call the P arm, and the other arm we call, I'm surprised she didn't wake up, I call one the P all the time. And the other arm, we call it the Q arm. So this happened on the Q arm of chromosome number 9, switched material with the Q arm of chromosome number 22. And then the numbers just mean the region. So I don't know how the regions are made, but maybe like this is, I don't know, there's 50 regions. It has to do with the banding. So some region number 34, oh, sorry, of the Q arm. Okay, so this is region number 34, switched with region number 11 of the Q arm of chromosome 22. That's the lingo, that's the nomenclature. And so, no, the chromosomes are not these beautiful fluorescent colors. This is a product of the computer software, but you can clearly see here the arrows are pointed here. After they arranged all the chromosomes, they said, like, oh man, chromosome 22 is so small. What happened? There's a deletion. Where did it, what happened to it? And so what they see is that then they look at chromosome 9. It's like, oh my gosh, what's this extra thing on chromosome 9? So they're able to see that there has been a translocation here. And so what ends up happening is that when people see this translocation, that person is either already has CML or is going to develop CML, which is pretty wild. So this is kind of what it looks like before they arrange it using a computer program. And still, the chromosomes are not going to be those different colors, but this is, I mean, I can tell these are homologous, but that's about it. So it's pretty, it's respectable, like it's really awesome career to be a genetic counselor. Okay, so last but not least, gametogenesis. And so this is basically the production of sperm or the production of eggs. And I just want to show one kind of overview because the terminology is pretty important, but there are some trends that you'll see. So remember that a germ cell is a 2N cell that will first undergo a bunch of mitosis to make a bunch of germ cells. And then when the time is right, those germ cells undergo meiosis to make gametes. Okay, so germ cells can also be called gametogonia. Okay, So I underline this part because this is the part that you can kind of see similar, no matter if you're talking about spermatogenesis or oogenesis. So people call it oogenesis, oogenesis. I don't know. I don't know the right way. I call it oogenesis. So these germ cells undergo mitosis, and then they're kind of, think of them kind of like the stem cells of gametes, right? So then they differentiate a little bit, and then they become gametocytes. Okay, so they're slightly different. They're still 2N, and these gametocytes are what actually undergo both phases of meiosis. Meiosis 1, remember at the end of meiosis 1, those cells are now haploid, but they're still pairs of sisters. Meiosis 2 happens, and so now the sisters are pulled apart. It's still haploid. And then sometimes you need more changes, so more differentiation, maturation needs to happen, and the end product are called gametes. These are 1N. Okay, so now let's look if we're talking about sperm. So the very, very early version, the germ cell version of sperm is called spermatogonia. See how that is still the same, spermatogonia. So spermatogonia, they develop a little bit, mature a little bit, differentiate a little bit. They're still 2N. They differentiate into what are called spermatocytes. Okay. Tocite. These spermatocytes, they don't look like sperm, they look like circles. They undergo meiosis 1 and meiosis 2, and here is something that is very important. They form, as all the cartoons we've shown so far, they form 2 circle cells, and the circles are about the same size. So we call that symmetrical division, right? Because each of the daughter cells, even though it's sperms, we still call them daughter cells, each of the daughter cells are approximately the same size, and they have a complete set of one complete set of chromosomes. So the product of symmetrical division, we call these things spermatids, kind of like sister chromatids. Okay, so we call them spermatids. Then these have to differentiate. They have to mature, they have to be ready for their final function, which is to function as sperm. So that's when the sperm goes from circles to actual little balloons, balloon-shaped tadpole-looking things. Those are called sperm. Okay. Here's a picture of it so you can see what's happening here. And so this process, once this spermatocyte, so remember, real quick, not the spermatogonia, those have already matured into these things called spermatocytes. Once the spermatocyte, it's time for it to undergo meiosis. It undergoes everything one phase after the next. Their phase of interkinesis. Remember, there's a phase of interkinesis right here. Right after telophase 1 and cytokinesis 1, you go into interkinesis. For sperm, they go right through it. There's no gap phase. If anything, they're there very briefly, but it's not very, very long time. So we'll just say extremely brief. And so for spermatogenesis, this happens continuously. Okay? No breaks, no pauses, no nothing. Just start it, finish the project. And it's symmetrical. So you can see symmetrical, what I mean, after meiosis, after anaphase 1, after, you can see here in telophase, both of those cells look about the same size. So if this is your starting size cell, then if we follow just this cell, this will undergo all the phases, meiosis 2, prophase 2, meiosis, metaphase 2, anaphase 2, telophase 2. These cells are also the same size. Same size. So this is symmetrical. Okay. Same size, symmetrical. Okay. So the spermatids have to undergo maturation and to become the sperm cells that are haploid. Okay, So continuous, symmetrical. And so this is crazy to me because it's so different from oogenesis that on a daily basis, a grown human male does this and produces several million spermatids a day. Several million, 10 to the 6 per day. Not 7 million, several. Okay, and this, if you think about it, not if you think about it, who can do this math in their brain, but if you calculated it, this ends up being about 1,500 spermatids per second, which is what? However, to go from spermatid to sperm cell, on average, 2 months. Weird. Okay, so 2 months to fully mature. Okay, so that's your kind of your overview of spermatogenesis. Nothing too wild. I mean, those stats are pretty crazy, but here's where the thing's gonna get even weirder. We're gonna go look at oogenesis. Okay, so I'm gonna go back to this chart. So we know this, we just covered spermatogonia all the way to sperm. Now let's just make sure the terminology, we're all on board with it. So the precursor, the germ cell, the very, very early baby stem cells that are gonna eventually become what are gonna be egg. They're called oogonia, okay? Just like spermatogonia, gametogonia, oogonia. Those undergo mitosis to build a huge pool of future eggs. Those future eggs are called oocytes, just like spermatocyte, gametocyte. Now these oocytes, once it's time for them to undergo meiosis, they do, but they don't form equally sized daughter cells. Because of that, we say that they undergo asymmetrical division. Right? So asymmetrical means different size daughter cells. Okay. What's all this other stuff I'm putting here? Pause? Yes. Okay. So when an oocyte undergoes meiosis, there's a long pause, not an interkinesis, in different phases. So there's checkpoints, and this pause can be really long. So the cell's like, okay, I literally just started meiosis. How long am I waiting to finish? Oh, I guess I'll just wait here until I'm told. Yeah. Not only is there one pause, there are 2 pauses. Crazily, even more, is because it's asymmetrical, you've got one cell that's big and giant, and then the other daughter cells are really tiny. So what we say is at the end of this, remember for sperm, if we go back up, you have 4 spermatids that look the same size. For oogenesis, you only have one round, big ass egg that's called an ootid. And then the 3 other ones that are really small, those are called polar bodies. So you have one ootid and 3 polar bodies. And so only this ootid becomes a viable egg called an ovum. Only one. So every time an oocyte undergoes meiosis, yes, it makes 4 gametes, but only one of them can be used to be fertilized. The other 3 get absorbed by the body. Let's take a look at this. Okay, so here we are. So for males, spermatogenesis happens at the start of puberty, and that's when it happens daily until it stops happening. Females, human females. The oocyte, so recall, this is already developed from the oogonia. The oogonia has undergone a bunch of mitosis to make oocytes, so we have an oocyte. Oocytes start meiosis in utero. When the female embryo is developing in the mother, in utero, a long time ago. Okay? So for everyone, years, for every female, years ago. Starts before birth. Then there's certain cues that force all of the oocytes. So oogonia make all of the oocytes that it's ever gonna make. It's gonna happen in utero. And then I had notes somewhere. Let me get it real quick. Yeah, okay. So before birth, 2 million. Yeah, I know that's not like several million per day, but still 2 million before birth. All 2 million of these oocytes start meiosis and then arrest, stop, pause in P1. All of them. So all of them are in the P1 phase, right? That's it. They wait until puberty starts. So they wait, I don't know, 12, 14 years. All of them are just hanging out in P1, prophase 1. However, along the way, a lot of them die. So by the time you get to puberty, now there's only about 60 to 80,000 remaining. Okay. So now 60 to 80,000, puberty, external cues, hormones, this is going to tell the cell, okay, you can go ahead and continue. So all of the cells, whatever remaining 60 to 80 of them, undergo metaphase 1, anaphase 1, telophase 1, cytokinesis 1, telophase and cytokinesis, here's your asymmetrical division. These are not the same size, different size. This is your asymmetrical division. This is going to be one polar body. So basically you can see that the cytoplasm, that means the cytosol and all of the organelles or most of the organelles gets pushed into one cell. And then the other polar body, the polar body still has the complete set, one complete set of chromosomes, but it really doesn't have the nutrients. It donated it to the future ootid. Because it's like, all right, ootid, I'm gonna give you all my stuff. So if you do get fertilized, you're gonna be able to provide a bunch of nutrients and a bunch of organelles and you're gonna get to go to work. Some cells like, yeah, I'm going through meiosis. Oh, what? I have to stop again? So all of these cells arrest in M2. So they go through metaphase, they start metaphase and they stop. So all of them kind of look like this. And then every 28 days, the cell gets another cue to continue. And so at this point, it's only one. Let me draw it a different color. Only one of these, one per month. So I guess it's one of the future ootids and one polar body. I'll call it future ootid. It's not a quite ootid yet, and one polar body continue. One per month, one per every 26 to 32 days. Okay. External cues, hormones. Levels go up, levels go down. That's when the cell knows, oh, time to continue. I'll go, we'll do it. And so then they go. So this cell here undergoes meiosis too. Okay. So it's starting to do all the things that we just talked about. This tiny little baby polar body undergoes the same meiosis, the same things we just talked about. And so this one here, so let's draw what it is, okay? So you have, let's say this is this cell. This is that cell, okay? And this big giant thing undergoes another asymmetrical division, asymmetrical. So that one of the cells, it's actually more like this, makes another big giant. It's more cytoplasm, more cytosol, more nutrients, more organelles. And then this little remaining part, I'll just draw like this. That's the other polar body. So at the end of meiosis too, you have 3 polar bodies. These are polar bodies, 3 of them. Okay. They all have the genetic material. They have a complete set, but they don't have the nutrients to exist. They've all donated it to the egg, sacrificed it to the egg. And so then at this point, this egg either becomes fertilized. Meanwhile, the uterus is starting to undergo a bunch of mitosis, building a really thick uterine wall. So either this egg, this ovum, either this gets fertilized and sits itself in that nice big thick uterine lining. If it doesn't, then the uterine lining undergoes apoptosis and all those cells that the uterus just made sheds and dies. And that's menstruation. So either this gets fertilized to become a 2N embryo, and that 2N embryo then undergoes mitosis to make a baby, to form a baby, or this does not get fertilized, it remains 1N, the uterine line sheds. And that's it. That's gametogenesis. So thank you for allowing me to go over. Not like a fair minute of you hearing this, but still. So that is the end of gametogenesis. Any questions? Okay. So hopefully that allows you to appreciate the differences between spermatogenesis and oogenesis and how the female body can do this properly every single time is crazy. Okay, so thank you. And I will see people, I think, Thursday if you show up. I hope you do. Spread the word. It's our last meeting. It'd be really awesome if we could maybe all just share our cameras and say hi, maybe. Okay. So I hope you have a great day, and I will see everyone soon.